Good afternoon. I'm Dr. Brian Shapiro, a marketing segment manager at ATCC. Thank you for joining us for the latest installment in the ATCC Excellence in Research webinar series. It's entitled Taming Uncertainty, Reducing Animal Byproducts in Cell Biology Workflows, and it's presented by Ms. Tiffany Cato. Ms. Cato is the Manager of Laboratory Operations at ATCC. In this presentation, Ms. Cato will discuss considerations around cell culture medium, focusing on a modified ABP-free McCoy's 5A medium formulation. She will then share data showing similar growth and viability characteristics of cells grown in both the original and the new media formulation. If you have any questions for our speaker, please use the chat function available through the webinar program. All questions will be answered as time allows at the end of the presentation. The recorded webinar presentation will be archived on the ATCC website, www.atcc.org. So with that, I'd like to welcome Tiffany Cato.
Thank you, Brian. Founded in 1925, ATCC is a nonprofit organization headquartered in Manassas, Virginia, with an R&D and services center in Gaithersburg, Maryland. ATCC is the world's premier biologics material resource and standards development organization. ATCC has over 5,000 cell lines and supporting media in our catalog, which leads me to today's webinar topic, reducing animal byproducts in cell biology workflows. Today we'll be discussing how to select cell biology media for workflows, including which critical attributes to take into account, as well as discussing considerations when using materials which contain animal byproducts. We'll also be discussing our current animal byproduct-free reagents before moving on to discussing the development of our newest basal medium, animal byproduct-free McCoy's 5A. So let's get started. Selection of cell culture reagents is often the first step in designing a new cell biology workflow. Whether you are in early discovery or ready to scale up your workflows, selection of cell culture media is critical for the success of your project. When making this important choice, there are several points to consider. ATCC provides recommendations for media formulations for the cell lines in our catalog. These recommendations are proven to support the growth and viability of ATCC cell lines. It is important to follow these recommendations. Failure to use ATCC recommended reagents may have negative impacts in cell health and viability, as well as leading to costly adoption and or rework. Media formulation may also vary from vendor to vendor and may cause osmotic stress to the cells if switched without adaptation. Another important factor to consider is serum. Most cell lines will require fetal bovine serum supplementation to support cell growth. It is critical to source well-characterized sera from locations such as Australia, the USA, or New Zealand. I'll discuss this point in more detail a bit further into this presentation. First, let's start by discussing basal media selection. Basal cell culture media are a complex mixture of salts, carbohydrates, vitamins, amino acids, metabolic precursors, growth factors, hormones, and trace elements. The requirements for these components vary among cell lines, and these differences are partly responsible for the extensive number of media formulations available on the market today. ATCC currently offers 9 classical basal media formulations. Our basal medias are optimized for the growth and scale-up of ATCC cell lines and are extensively tested to support cell growth for each lot. So how do you know which basal media to use for your cell line? Well, if you're working with an ATCC cell line, it's easy. It is the recommended media on the product sheet on the cell line website. This media will have been validated to work with the specific cell line in question, but if you're starting from scratch, it can be a bit more difficult. Each basal media has its own unique properties, which are ideal for certain cell types. Let's start with EMEM. This medium is one of the first widely used media for cell culture and was formulated by Harry Eagle from his earlier and simpler basal media, simply called BME. EMEM was originally formulated to use with mouse L cells and HeLa cells. Since it's a rather simple formulation, it typically requires additional supplementation or higher levels of serum, typically 10% or greater. The next two mediums we have are modifications of EMEM, starting with DMEM. This is a modification of EMEM with roughly twice the concentration of amino acids and 4 times the amount of vitamins as EMEM. In addition to that, DMEM has ferric nitrate, sodium pyruvate, and some supplementary amino acids. ATCC DMEM also contains an increased concentration of glucose to help support cell growth. The additional nutrients in this medium means that it can support a wide variety of cell types with various concentrations of serum supplementation. IMDM is a further modification of DMEM. Compared to DMEM, IMDM has additional amino acids, vitamins, and inorganic salts. Potassium nitrate was substituted for ferric nitrate in this formulation. This medium is really great for supporting the growth of suspension cell lines as it was originally designed for the growth of lymphocytes and hybridomas. For cell lines which have high protein productions, like hybridomas, it may require additional supplementation with L-glutamine. HAMS F12K is a modification of the original HAMS medium. HAMS medium was designed to support the growth of CHO cells, Chinese hamster ovary cells. The CONGS modification has increased amounts of amino acids, pyruvate, biotin, calcium, magnesium, putrescin, and phenol red. This rich medium formulation is designed to support the growth and differentiation of primary cells with or without serum. Continuing to look at basal media formulations, we have DMEM F12. This medium is a 1-to-1 mixture of DMEM and HAMS F12. It's extremely rich and complex media, and it'll support the growth of a broad range of cell types in both serum and serum-free formulations. McCoy's 5A and RPMI 1640 were developed at Roswell Park Memorial Institute in Buffalo, New York. McCoy's 5A was originally used to grow Novikov hepatoma cells. McCoy's contains bactopeptone, which is a commercially available supplement that is composed of an animal origin enzymatic digest of bovine and porcine animal proteins. This acts as an additional nitrogen source in the culture medium. This is the only basal medium in the ATCC catalog which contains animal byproduct. RPMI 1640 is a modification of McCoy's 5A and was developed for the long-term culture of lymphocytes. RPMI 1640 will support the growth of a wide variety of cells in suspension, as well as a number of cells grown as monolayers. And finally, we have L15 medium. L15 medium is formulated for use without CO2 and is commonly used to support the growth of insect, fish, and human cell lines. Primary cells often require complex media to support the establishment of cultures. This medium and the corresponding growth kits are tailored to the specific tissue and cell type. For primary cells, there are several growth mediums available. These are specifically formulated with growth factors, cytokines, and vitamins. These can be largely categorized by the cell type they are designed to support. Starting first with endothelial cells. These cells form the endothelium, the thin layer of cells that line the interior surface of blood vessels. Endothelial cells can be cultured in complete growth media containing either bovine brain extract, BBE, or vascular endothelial growth factor, VGF or VEGF. Use of the endothelial cell growth kit with VEGF will support a faster rate of proliferation, while the endothelial cell growth kit with BBE is recommended if a less desired cell culture medium is wanted. Smooth muscle cells originate from involuntary non-striated muscles, such as vascular smooth muscle, lung smooth muscle, and bladder smooth muscle. The basal media and corresponding growth kit are designed to create a complete cell culture environment for these cells. Next, we have epithelial cells. These cells line the cavities of glands and organs in the body and also cover flat surfaces such as the skin. Given the diversity of tissues in which these cells are found, there are a similarly diverse offering of basal medias and growth kits tailored to the tissue of origin for these cells. So next we have fibroblasts. These cells are critical to maintain the structural integrity of connective tissue and synthesize extracellular matrix proteins. These are often fast-growing cells and can be grown in low serum or, for more defined media, serum-free conditions. And you can see we have both options as growth kits to the right. Keratinocytes are the most common type of skin cell. They make up the majority of the epidermis, and they produce keratin. Most keratinocytes in primary cell culture have been isolated from the epidermis of human juvenile foreskin or adult skin. Keratinocytes cultured in serum-free, low-calcium growth medium will maintain the cells in an undifferentiated, proliferative state while inhibiting fibroblast outgrowth. Another cell type of the epidermis are melanocytes. They are specialized skin cells that produce the pigment melanin, which gives skin its color and protects it from the hazardous effects of UV radiation. Melanocytes for research are isolated from the epidermis of human juvenile foreskin or human adult skin. Depending on the source, there are two options for growth kits for melanocytes. And finally, my favorite, we have mesenchymal stem cells. These are self-renewing, multipotent adult stem cells, also known as MSCs. MSCs are traditionally found in the bone marrow, but can also be isolated from other tissues. This includes adipose tissue, human umbilical cord, and peripheral blood. These cells can be differentiated into osteocytes, chondrocytes, and adipocytes. ATCC offers growth kits to support MSC growth, as well as differentiation kits. These kits have been optimized, which is really great. It reduces the time spent acquiring materials and optimizing your differentiation protocols and makes for a great time saver. All right, now that we've discussed basal media and primary cell growth kits, let's dig into another point of consideration when selecting reagents for your cell biology workflow: animal byproducts. Animal byproducts are derived from the blood or body of animals. Common animal byproducts used in cell culture are fetal bovine serum, extracellular matrix, and some growth factors. There's a few issues with using animal byproducts, including, and most often critically, the potential introduction of adventitious agents. Adventitious agents are defined by the World Health Organization as contaminating microorganisms of the cell culture or source material. Adventitious agents can be introduced through cell lines which are contaminated, through animal byproducts, through the environment, specifically failure to maintain aseptic or sterile environment, and through the direct use of animals in bioprocesses. This last point refers to processes like influenza culturing eggs, where the animal is directly used in the process. This is out of the scope of today's discussion. So how do you minimize the risk of introducing adventitious agents into your workflows? Well, there's a few ways. First and foremost, use cell lines that are well characterized from reputable sources. Test your cell culture and process and post-banking for contamination. Avoid animal byproducts whenever possible and as early as possible when designing your workflow. And finally, when you can't avoid using animal byproducts, use sera and reagents that are tested according to 9 CFR 113.53. And we'll discuss this regulation in more detail in a bit. So to start, you absolutely need to know your cells. This means knowing that your cells are what they purport to be and whether or not they harbor bacteria or viruses. For human and mouse cell lines, identity of the individual cell line can be confirmed through short tandem repeat testing. This is a capillary electrophoresis assay, which analyzes the frequency of allele repeats at specific loci. This establishes a unique DNA fingerprint for every cell line. This is performed for every human cell line at ATCC and is offered as a service for human and mouse cell lines. Check out atcc.org/STR to learn more about this service. For cell lines which are not human or mouse, the identity of these cell lines can be verified to the species level. This is done using a PCR test to amplify the cytochrome oxidase 1 gene. And this allows for determination of species and gives confidence that the cells you're working with are the expected species and are not cross-contaminated with another species. As for microbial contamination, bacterial contamination testing is performed for both aerobic and anaerobic bacteria, as well as viral testing for HIV, Hep B, HPV, EBV, and CMV in all human cell lines produced at ATCC. So while we're on the subject of microbial contamination, let's talk about mycoplasma. Nobody likes it, but we're going to talk about it. This is a common bacterial contaminant in cell culture that often goes undetected. The reason mycoplasma contamination is so difficult to detect is that they are extremely small bacterial cells with no cell wall, and you cannot observe them through a microscope. These bacteria do not cause media to become turbid or acidic, and these tiny organisms can have huge impacts on research. They can inhibit cell growth, they can disrupt nucleic acid synthesis, and they can stimulate stress response pathways in your cells, which can impact your experimental results. So how can we detect these if we can't see them? PCR is widely accepted as a method for detection. ATCC offers a PCR kit as well as a sample submission service where we do the testing for you. The service sample submission process is quite painless. Once you order, ATCC will send you a Whatman FTA sample collection card. You spot your sample on this card and you send it back to us. ATCC experts will process your sample, analyze the result, and send you a comprehensive report within 3 to 5 business days. It's recommended to test early and often for mycoplasma contamination. Test when you receive your cell line, test after 10 passages, after preparing a cell bank, and simply when in doubt. If you prefer a more do-it-yourself approach, the Universal Mycoplasma Detection Kit might be your preferred option. This is an easy-to-use kit that includes all the components needed for the PCR reaction and comes pre-optimized for the detection of over 60 species of mycoplasma. Each kit contains enough material to perform 40 assays. All right, so now that we've discussed using authenticated and characterized cell lines, let's talk a little bit about fetal bovine serum. Serum is a pretty critical component in cell culture and often cannot be removed or substituted. In cases where the serum cannot be removed or substituted, it's important to use serum that is extensively tested according to Title IX CFR 113.53. This is a government guideline from the Animal and Plant Health Inspection Service, also known as APHIS. This requires serum to be tested using a 21-day in vitro test for viruses. In addition to this testing, it is also recommended to use serum that is tested for bacteria and fungi in compliance with USP recommendations. Endotoxin testing is also critical. High levels of endotoxin may activate immune responses in your cell culture and lead to undesirable effects. ATC serum is tested for all of these, as well as triple filtered with 0.1 micron filters. Each lot is additionally tested for growth and proliferation for several cell lines. Additionally, ATC serum is collected in USDA inspected abattoirs. United States serum reduces the risk of bovine spongiform encephalopathy, also known as BSE, As opposed to other geographical locations where BSE is or has been endemic, USA sourced fetal bovine has reduced risk of BSE. And of course, the best course of action is to reduce the potential of adventitious agents through the use of chemically defined animal byproduct-free reagents. ATCC currently offers several animal byproduct-free solutions for cell biology. This includes reagents for cryopreservation, such as stem cell freezing medium and serum-free freezing medium. For cell growth and expansion workflows, there are dissociation reagents, such as stem cell dissociation reagent, non-enzymatic cell dissociation solution, and to halt the dissociation, there is soybean trypsin inhibitor. For growth, all of ATCC basal mediums are animal byproduct free, with the exception of McCoy's 5A. However, ATCC has reformulated McCoy's 5A to remove the bactopeptone, therefore making this medium animal byproduct free as well. As I just mentioned, we are excited to announce our reformulation of McCoy's 5A. The new formulation is the same as the previous, but with the removal of bactopeptone. This new medium is being called McCoy's 5A medium, ABP-free, with catalog number 30-2011. This new formulation has quite a few added benefits compared to the previous formulation, such as reduced risk to process and product due to the removal of animal byproducts. The new formulation does not require adoption from the previous formulation. The new formulation has been proven to support cell health and growth of cells grown in the previous McCoy's formulation. And finally, ATCC will be remanufacturing all of our cell lines grown in the previous McCoy's formulation in the new animal byproduct-free McCoy's formulation for a full end-to-end solution. So let's talk about the development of this new medium. This medium is identical to the previous McCoy's 5A formulation, with the exception that it does not have bactopeptone, therefore making it animal byproduct free. ATCC extensively tested the performance of this new formulation against the previous formulation. There is a two-pronged approach to the performance evaluation of the animal byproduct-free McCoys. The first portion of the evaluation focused on the short-term proliferation and potential cytotoxic effects of the new formulation when compared against the old formulation. For this, we used 3 cell lines, which are typically grown in the previous McCoy's 5A formulation. These cell lines were selected based on their similar growth rates and ability to produce uniform monolayers. And for this, HCT116, MESOV, HT29 were selected. To measure cell growth, cells were plated in 96 well plates and monitored for 48 to 96 hours. Cell proliferation was measured using the Excelligence RTCA system by Agilent. The Excelligence RTA system utilizes electrical impedance as a tool to evaluate real-time proliferation, cell number, size, and shape. This impedance is reported by the instrument software as cell index. So the higher the cell index, the more cells impeding the electric signal. As you can see on the figure to the left, all 3 cell lines proliferated at similar rates in both the old McCoys and the new animal byproduct-free McCoys. The new animal byproduct-free is shown in light purple, whereas the old McCoys is shown in dark purple. And these results indicate that the new formulation provided all of the needed nutrients for the growth of the cells without the needed addition of bactopeptone. The second portion of the short-term test was a cell viability assay. We performed a cell viability assay using the cell titer glow assay by Promega. The purpose of this assay was to measure ATP levels as a means of determining viability and to measure potential cytotoxic effects of the new media formulation. The cell titer glow assay is a single reagent assay that uses luciferase to quantify ATP. The resulting luminescence values are proportional to the percentage of metabolically active viable cells present. Cells were thawed directly in complete culture media prepared with the animal byproduct-free McCoys alongside the old McCoys. And this was done without an adaptation period. Cells were seeded in such a way again to reach confluence within 48 to 96 hours, and each experiment was performed in triplicate for both the proliferation and the viability assays. And again, looking at the figure on the left, light purple is the new McCoys, dark purple is the old McCoys. We can see the RL values for both conditions are very similar, which shows again the new McCoys support cell health and viability in a manner similar to the previous McCoys formulation. So once we had the short-term experiments done, we wanted to move on to a long-term growth experiments. This experiment was crucial as most labs maintain and expand cells for weeks at a time, and it's important to understand the potential impact of the new formulation. In this study, our most popular cell lines grown in McCoy's medium were selected, with the exception of Kasumi 3, which was selected based on its similarity of the cell line used to develop the original McCoy's 5A formulation. The 5 cell lines selected were HCT116, MESOV, HT29, SKES1, and as mentioned before, Kasumi 3. In this study, cell cultures were maintained according to established procedures at ATCC for 21 to 23 days. Cells were thawed directly into complete culture media prepared with McCoy's 5A animal byproduct free without an adaptation period, alongside control cell media, which was the previous McCoy's formulation. Cells were maintained in log phase growth throughout the evaluation through regular subculturing in tissue culture flasks. Population doubling time and viability were recorded at each subculture. The averages of these values were then compared between the two conditions. As you can see in the figure, the doubling times were roughly equivalent for all cell lines with the exception of Kasumi 3, which actually performed better in the new McCoy's formulation. This data shows that, again, the new McCoy's provides the needed nutrients to support cell growth, and furthermore shows an adaptation period is not needed to switch between the old McCoy's formulation and the new animal byproduct-free formulation. Here we see additional data for the long-term study. At each subculture, viability was measured. This was measured using an automated trypan blue exclusion method. The average percent viability of cells grown in McCoy's 5A medium ABP-free was compared to those grown in McCoy's 5A medium. As you can see, the new McCoy's formulation supported cell viabilities above 80% for all cell lines and performed equivalently to the old McCoy's formulation. This data clearly shows that the new animal byproduct-free McCoy's is a suitable alternative to the old McCoy's and is an excellent addition to the ATCC basal culture medium portfolio. Our next steps in supporting the scientific community and our European Union-based customers is to remanufacture all of our McCoy's cell lines in the new animal byproduct-free McCoy's formulations. These newly remanufactured items will comply with the changing regulatory landscape in the European Union. As of today, we have relaunched 4 cell lines with a 5th coming soon this year. The relaunched cell lines are MES-SA, ME180, HEC1A, MES-SA/MX, and coming soon is MALM3, MALME3. So in conclusion, animal byproducts should be avoided when selecting cell biology reagents when possible. ATCC offers several animal byproduct-free solutions, and we are expanding our offerings with the new McCoy's 5A animal byproduct-free formulation. ATCC is remanufacturing its McCoy's media cell lines in this xeno-free version to better support the scientific community in navigating the safety and regulatory challenges associated with animal byproduct containing cell lines. ATCC is collecting additional NGS data in support of these new offerings to be available to customers beginning in Q3. This data will provide key comparison data for metabolic pathway activity between cells grown in old McCoys and the new animal byproduct-free McCoys. We're hopeful this will provide our customers with critical data to support their switch from the old McCoy's formulation to the new McCoy's formulation. Thank you for your time today.
All righty. Well, thank you, Tiffany, for that excellent presentation. In just a few moments, we will begin our Q&A session. Please be sure and use the chat function available through the webinar program to submit your questions. The recorded webinar presentation will be available on demand on the ATCC website at www.atcc.org. All right, well, let's go ahead and jump into our first question. We've had quite a few come in so far. So, Tiffany, does the new McCoy's 5A ABP-free medium still need to be supplemented with fetal bovine serum?
Thanks, Brian. The new McCoy's formulation is the same as the previous formulation, except for the removal of the bactopeptone. So that means it does still need to be supplemented with serum.
All right. Now, this is somewhat of a follow-up question. Will all of the ATCC cell lines grown in McCoy's 5A be remade in the new animal byproduct-free, bactopeptone-free formulation?
Yeah, so that's the plan. We're going to be selling our current stock, and as we restock, we will be manufacturing those new cell banks in the animal byproduct-free formulation.
Great, great. And I think you might have mentioned this in your presentation, but can you grow the new reformulated McCoy cell lines in the old McCoy's formulation?
Yep. So the cell lines are compatible with both formulations. The data indicates that you don't need a transition period, so you can switch between the two formulations as desired.
Excellent, excellent. All right, now there's been this great movement towards sequencing data around cell lines, and ATCC actually has a sequencing and bioinformatics core that's cranking out the sequences of many of our organisms, whole genomic for prokaryotic, transcriptomic, RNA-seq, that sort of thing for eukaryotic. Have we done any sequencing on the cells that are specially made to be grown in the reformulated new McCoy's medium?
Yeah, so we're really spoiled with our bioinformatics system, and we are going to be using that resource to get an RNA-seq panel. We're going to look at some critical metabolic pathways, and we're going to have comparison data. So you'll see the data of the cell line grown in both formulations. So if you need to switch from one to the other and you have regulatory burdens, this is another piece of information to support that switch.
Nice, nice. Now, how do I know if the cell line has been made in the new McCoy's formulation?
Yeah, that one's pretty easy. So if you're on the ATCC website, you'll notice the cell lines have a name. This usually looks like HCT116, but they also have a catalog number that is over there on, it's usually on the header, big and bold. It'll look like CRL123. If you see that it's CRL123.NM, it means new McCoy's. So if you see that .NM, it's been made in the new McCoy's. If there is no .NM, that means it was likely made in the old McCoy's or a completely different media that's not McCoy's.
All right, good answer, good answer. Now, will both McCoy's formulations be available for sale?
Yep, you can purchase both formulations on the website today.
Great, great. And where can I find more information about the new McCoy's 5A study that you presented?
Yeah, so if you go on the ATCC website and you search for catalog number 30-2011, you'll find the product page for the new McCoy's. On the right-hand side, there is a downloadable application note. It has all of the data. It's about 2 pages long, so you can download that for your records.
All right, great. Now, this is a trick question someone wanted to throw at you. If I still have to supplement with fetal bovine serum, what's the benefit of the animal byproduct-free McCoys?
Yeah, that's a really good question. So the benefit is that you're still reducing the overall risk of introducing adventitious agents. Instead of having 2 sources of animal byproducts in your system, you've eliminated one. So now you have your serum, and serum is becoming more and more better qualified. There's a lot of regulations around it. So it's a very defined animal byproduct. Hopefully, eventually in 20 years, we'll all be animal byproduct free, but as we get there, this is the first step forward.
Nice. All right. We've had a bunch of questions come in that are specific to mycoplasma, and then some that are general around cell biology that we'll address a few of those now. So, Tiffany, this is around the mycoplasma detection kit, actually. Can you identify the mycoplasma species with the mycoplasma detection kit?
Yeah, so with the kit, it's more of a identification of contamination being present. You'll get a band if there is one of the 60 species that are detectable, amplifiable by the primers. So it'll tell you if you do have the contamination or you don't. It won't tell you which species, and it is very specific to mycoplasma. So you won't get bands if you have other bacteria or microbes in your sample. It's very specifically mycoplasma.
Great. Now, this next question, we actually get in quite a bit. And it's, can you use a contaminated culture in your experiments, presumably, after you've treated them with antibiotics?
So generally speaking, it's not recommended to use cells that have been contaminated. In addition, for the contamination being difficult to truly get rid of, there may be changes to the cells through the stress of the contamination and subsequent curing with antibiotics. It's better to go back to a clean bank if you have that available.
All right, good. Here's a great question. What could be a reason for my cells unexpectedly detaching from the plate?
Yeah, so there's a few reasons this can happen. Most often it's due to suboptimal media pH and incorrect buffering. This can be from an imbalance of the percent of CO2 in the incubator and the amount of sodium bicarb in the basal media. Most of the ATCC formulated basal medias contain 1.5 grams per liter of bicarb, which is optimized for 5% CO2 environments. If you're using an alternative DMEM that contains 3.7 grams per liter of bicarbonate, the cells should be incubated in 10% CO2. Increased CO2 is necessary when you're using that higher level of sodium bicarb in order for the buffering to work as appropriate. Cell detachment can also be caused by the confluency and density of the cells in the flask. If you allow your cultures to become overconfluent, they will start lifting off, sloughing off. May also be used to describe this, just because there's too many. And then finally, the concentration and the type of exposure to the dissociation enzymes used on the cells during subculture. If you don't properly inactivate the dissociation agent, your cells will fail to reattach, and you can get clumping cells, and those clumping cells may not reattach nicely and can easily lift off after subculture.
All right, great. Now, here's another question about the mycoplasma detection kit. How many samples can you run with that?
Yeah, so there's 40 reactions in the kit, but we recommend that you run about 12 samples, and that gives you triplicates for each sample. So you can rest assured that your results are definitely certain. You'll have double redundancy. So yeah, 12.
All right. Wow, we've had a bunch more questions come in. This first one is around cons media. And it's really an optimization question. Would it work for any primary cell in the human body? I mean, is it something that we've internally optimized for work in primary liver or heart cells? Which I do know that at the very moment we don't have primary liver cells at ATCC.
Yeah, so cons medium is really rich. You can likely use it for many cell types. As for optimizing specifically for heart or liver, I don't specifically have experience. Our tech services may have more information, so definitely reach out to them and see if they've got recommendations.
Nice. All right, and this is probably something that we haven't done, but have we done a comparison of our endothelial cell medium with Lonza's? Are they comparable in growth dynamics?
I'm sure both kits have been optimized to support the growth of endothelial, but we have not specifically compared the Lonza brand against the ATCC brand.
Okay, fair enough. And then this question, are your cryopreserved reagents DMSO-free? And that's probably specific to the reagent itself, right? And we can have a technical service rep reach out to you with a specific answer around that. All right, moving on to the next question. It's about co-culturing cells. Given the complexity of co-culturing two distinct cell types such as primary macrophages and CD3 positive T cells, what kind of considerations or adjustments would you recommend in formulating cell culture media around the compatibility and optimal performance of both cell types? And what kind of considerations might impact media formulation nutrients? It's a big question.
Yeah, so co-culturing can be really tricky. You got to make both cell types happy and not introduce additional variation to your experiment, especially the one listed here. Both cell types are really sensitive to stimulation. So typically, in my experience, you grow the cells in the defined media needed for each cell type separately. And as you approach the actual co-culture point, you kind of take off those additional additives, the stimulants, and you go to a more simple, basic media. It's going to be required to be optimized, and especially if you're using primary T cells, it's going to be donor to donor, the performance. Some are going to be really tricky. Some might be a little bit steady eddy. So all of that to say, it depends.
Yeah, that's a great answer. I mean, it sounds like there's some empirical experimentation that needs to be done really to optimize your assay. And the best place to look is the literature. Who's done it before, right?
Yep, absolutely. And it honestly just requires time on the bench and running a bunch of iterations to get that perfect formulation. But usually the literature will give you a good launching point to get your formulation nailed down.
I love it. All right. Okay, this next question is kind of loaded. What's the outlook for animal byproduct-free FBS? Well, FBS comes from an animal, right? So I don't know about that. We do have some media or some antibiotic-free or more defined mediums like for iPSCs, right?
Yeah, our iPSC systems are largely animal byproduct-free except for the extracellular matrix, of course. As for other additives as FBS, we currently don't offer alternatives. I think ATCC, like the rest of the industry, is always looking forward and are hopeful to the day that we do get rid of serum, but I would not count on it, certainly not next year, not in the next few years, unfortunately.
All right. No, that's a good answer. That's a good answer to that. All right, and the last question that we have, so I'll make this last call while I'm asking this. If any more come in, we can address or we can close. But can I keep wanting to say the wrong things for ABP, but animal byproduct-free medium, can it aid in preventing cell senescence?
That's a good question. So I wouldn't say necessarily that it's going to aid in preventing senescence. However, it's chemically defined. So you're going to get really regular performance out of animal byproduct-free media. So depending on what you're doing, if you're doing primary cells, you'll see a very reliable result. Of course, your donor is going to introduce variability, but your cells aren't going to be senescing because one lot of serum was not as good as the other necessarily, or one lot of McCoy's media was better than another. So in that sense, it can help you get more reliable results particularly around senescence.
All righty. Well, at this time, we will conclude our Q&A session. I'd like to thank Tiffany for the excellent presentation, and thank you everyone for attending this webinar. If you're interested in ordering ATCC's ABP-free medium, please visit our website at www.atcc.org. Thank you again, everyone, and have a great day.